Good afternoon, everyone. First of all, I'd like to thank Norberto for inviting me to give this talk here. Such a nice meeting, such a nice audience. So this is a topic that you may know better than me. I'm not a neuroanatomist, I'm just a physicist. But you know that temporal lobe epilepsy, people who have, or subjects who have temporal lobe epilepsy, they have a large restructuring of the dentate gyrus, which includes lots of features, including sclerosis, which is, let's see if I can point it over there, characterized by lots of structural rearrangements in the network of the dentate gyrus. The most conspicuous one is sprouting of mossy fibers, loss of Hiller cells, but there's also hectopic positioning of granule cells. There's morphological alterations in granule cells. So there are lots of structural and morphological alterations in the dentate gyrus in subjects who undergo seizures, repeated seizures, in the case of temporal lobe epilepsy. And it's well known that this mossy fiber sproutings, I hope you can see them there, which usually project to the CA3 area of hippocampus. In epileptic patients, they project back to the dentate gyrus and innervate the granule cells themselves, creating a recurrent, I cannot point from here, but creating a recurrent circuit. This may have some consequences for the epilepsy of the subject. You also may know that Ivan Soltesh in California, with people from his lab, they have constructed a model, or should I say a series of models, computational models for the dentate gyrus with different levels of complexity, starting with models of one over 2000 scale, 1 to 20, and even 1-to-1 scale, which they just use it for structural research, containing lots of cell models, 500 neurons at 50,000 cells, to study the role that structural alterations in the dentate gyrus have on the hyperexcitability of the network. So this slide here shows the structure of the network, the pattern of connectivity that they have in their model. So they have basically 4 neuron types in the model: the granule cells, the mossy cells, the basket cells, and the hip cells, hippocampal inhibitory interneurons, which project to the perforant path afferents. You can see the projections in the slide over there. You can see, here you can see the 4 neurons, and there you can see the network. This is really awkward to do this, but okay, I hope you can see that. The basic strategy that they use in their modeling is to simulate focal stimulation of the dentate from the perforant path by simulating, say, a group of 100 adjacent neurons, adjacent granule cells in the network. And then after the finishing of the simulation, they let the system evolve and evaluate the excitability of this system. And they test what happens when they introduce sprouted mossy fibers, which you can see in black. Somewhere over there. I cannot see from here, so I can see my screen, but not from there. Anyhow, you can see the black line joining the granule cell with itself in this slide. So they basically constructed a network model using these 4 neuron models to study the role of topology, basically, the structure of the network on epilepsy, on the hyperexcitability of the network. They've been doing this since 2005. when Norberto talked about the collaboration, that this area of computational neuroscience urges us to create bonds, to create collaborations between us, to share our models, our thoughts, our investigations. People are really active in the field of modeling. There are resources like ModelDB, which I'm just showing here, in which people who create models, could be neuronal models, or channel models, or network models. They just deposit them there. They make it then to make the models publicly available to anyone who wants to investigate this model. So this is the model constructed by Soltesh and his collaborators. The models are just available at the site here for anyone to download and plug it into their computer. And well, they use Neuron as the simulator, so you have to you need to have Neuron in your computer, which also is freely available, so you can enter the main site and download it and start using it. So going back to the story. Soltesh, what they use in the models are so-called reduced neuron models, which are, from the point of view of neuroanatomists, kind of poor, morphologically poor models of the cells. They contain all the ionic channels present in those cells, but the structure of the models are much more simplified in comparison to the real ones. So here you see the 4 neural models that they use in the network models. You see that they are simplified. The word reduced means that they have a small number of compartments, which means that actually they have a small number of parameters, of variables, which also means that it's cheaper to simulate than in computer, but they may be losing some features of the neurons, which may be relevant for the function that they have. In this work here, basically, we're going to concentrate on the granule cell. So you see the granule cell there is basically constituted of a soma and 2 dendritic branches with 3 compartments per branch. Coming back to the story, so you got the idea. He has this toy model, this network model, made of those 4 neuron types, and the network model has, the simpler one has 500 neurons, the more complicated one has 50,000 neurons. And they simulating focal stimulation of coming from the performance path, and they evaluate the activity of the network, for example, using the RASI plots that I show here. So you can see, in the case of, in the top line, you have the normal network without sprouting. So you see that just this excitation there, and then the activity dies out instantaneously after the end of stimulus. Then with the 10% sprouting, you see this, the propagation of the excitation throughout the dentate model. It's important to say that dentate model is a ring, has a ring-like structure. So it's closed. It's not like the real dentate from the hippocampus, but usually people who run simulations, they do that to avoid border effects. So the last neuron, say the 500th neuron in the network is neighbor of the first neuron. So you don't need, you don't have border effects in this kind of structure. So that's why you have this structure shown there. So these neurons there are neighbors of neurons there. So everything is closed. So what this raster means is that excitation is propagating throughout the network, from the point where a stimulus arrives towards the other end of the network. But then it dies out after some 200 milliseconds. And the thing goes on for 25% of sprouting, 50% of sprouting, and you can see that as you increase the percentage of sprouting in the network, you reach a state in which the activity is self-sustained. So it's a kind of epileptic, meaning that the network became hyper-excitable. And when they did that, say with 10% sprouting, which is the kind of just borderline state, and increase the percentage of mostly cell loss, they didn't observe any major change in the network behavior with respect to activity. In terms of hyperexcitability, what they observed is that activity is interrupted after some milliseconds, which meaning that the cell loss, mostly cell loss or healer cell loss, has the effect of has an effect the possibility of interrupting or reducing the hyperexcitability of the system. But the major conclusion from their work is that the most critical factor for excitability of the dentate gyrus is mostly fibrous sprouting, at least from the topological point of view, rather than the mostly cell loss. And the way they explain that is basically on topology. We've been talking about network topology in this session since the first talk by Norberto. So you are aware of the concepts of clustering coefficient and average path length. What happens in the dentate gyrus is that here you have the healthy one and the one with sclerosis, in which you have mostly fiber sprouting and you have healer cell loss. So you have some sprouts there. You lose some, actually you create some connections there, and you have you lose some of those mostly cells that are responsible for creating long-range connections. But you still keep some of them. The important thing is that you still have some of the connections there. Yeah, maybe more. Can't you hear me in the back? No? Even if I shout? Okay, I'm sorry about that. So, well. Okay. Maybe. I don't like to talk in place like I prefer to walk and make justice. Okay. So you see here, you have the healthy network, the network simulating a healthy subject and the network for a subject with sclerosis. What they found is that when you have a healthy network has already a small word structure, which has a high clustering coefficient because you have, you have some connections between neighboring neurons, and also a small path length. So you can go from one node of the network to another one in a few synapses via the long-range connections provided by the mostly cells. When you have sclerosis, you have sprouting, you enhance the small word characteristic of the network. So you increase the clustering of the network by the creation of those local connections provided by the sproutings. And although you lose some of the long-range connections provided by the mostly cells, you keep some of them. So the path length is still more or less the same as it was before, but the clustering coefficient increases. So the explanation they give for the increase of excitability of the network is that the sclerotic network has a stronger small word structure. The small wordliness, let's say, a structure of the network is increased, is favored, is enhanced. And this leads to hyperexcitability and epilepsy. Based on when I entered in this area via the collaboration with Norberto, I discovered that there are more changes in the dentate gyrus in an epileptic subject than the structural, than the topological changes that are known from the work of Soltes. In particular, Norberto and his collaborators have shown that granule cells have, the newborn granule cells, dentate gyrus is one of the regions in the brain where you have birth of new neurons. throughout life. So some of those new cells in rats that undergo status epilepticus induced by pilocarpine, they have morphological changes, morphological alterations, which correspond to this case here. You can observe, you can compare with the control case, which is just a newborn cell from a rat that doesn't undergo status epilepticus, and it has the normal structure of a granule cell. The one from a pelo-treated animal has a shorter dendritic arborization with more branches close to the soma, close to the inner molecular layer. Norberto and Gabriel, one of his students, they reconstructed, they morphologically reconstructed those cells using NEWroscience data. And they deposit those reconstructions in neuromorph.org, which is another database of data, of neuronal data, publicly available for anyone who wants to construct network or neuronal models. And let's go back to that slide. And based on the models, which are only anatomical reconstructions, only morphological reconstructions, we in our lab we constructed not reduced but say full neuronal models of those cells, which are shown. I mean, I just showed two examples of them here. One of a control cell, one of a cell from a pelo-treated animal. So you can see that the number of compartments making the dendrites, the dendritic arbor, is much larger than in the original model of Soltes. And we distributed throughout those dendrites the ionic channels that are known to exist in granule cells, with their densities, the densities of the maximum conductances, which are needed for a modeler to construct a biophysical, we call realistic model of those cells. We had a problem here, which is nobody knows how is the distribution of the maximum conductances of those ionic channels in a newborn, morphologically altered granule cell. Nobody knows that because people in, say, physiology didn't do this already. People know what's the distribution in the control, in the case of normal cells, but they don't know how they are distributed in the case of pelo cells, cells from pelo-treated animals. So what we did in our study, which was designed to single out just the effect of morphology, we just wanted to see what morphology would generate in the cells. We kept the same densities. We assumed that the ionic channels are the same, which is reasonable, but also the densities, which means that the maximum conductances of all those channels in each area, in each part of the proximal dendrite, medial dendrites, distal dendrites of the cell, they are the same. They are the same ones observed, found out experimentally in the control cells. This is a weak point of our research, but we don't have any other option in that case. And it makes sense in our say global design, which is to study only the morphological effects of those changes. So this is one point you need to have in mind, that we have the densities of the ionic channels the same in both cases. Only the morphology changes. So, and then what we did is to study the effect of the morphology change in single cell models. So we basically we reconstructed cell models for a population of cells detected by Norberto and his students, and we simulated inputs arriving to those cells in an epileptic animal. In particular, we simulated the mossy fiber inputs that arrive in the molecular layer, and all the inputs that arrive in other parts of the cell by simulating electrodes placed in different areas of those cells. In this slide here, I only show some examples, but we simulated different types of inputs arriving in different areas of each single cell model in our database. And what we observed is that newborn cells with altered morphology are less excitable than the control ones. So you can see here, these are the inputs used, constant current injections with certain durations. And let's go back to here. It's better for me to watch in this screen here. So you see here in green the control spikes, and in orange the spikes of the pelo-treated cell. And you see that the frequency of those spikes is much smaller than in the case of in the control case. You also can see here in the bottom figure, the bottom graph, the frequency against current, Fi as we call curve, showing that the frequency for pelo-treated animals is systematically lower than the frequency for control animals in our simulations. So based on our single cell simulations, we could predict that the morphological alterations in those granule cells, in those newborn granule cells, could have a sort of protective effect on the network because they become less excitable. So this excited us to carry on our work and construct a network model of the entire dentate gyrus, of course a scaled down model, containing those morphologically altered cells to see the effect of those cells in the network context. So not to reinvent the wheel, we started by using the same model that Soltes and his collaborators used. So we took the 1 to 2000 scale version of the model, which is the simplest one, the less expensive one to be run in our computers, which is a model which is already large for computational neuroscience point of view. It has 512 neurons. And we replaced all the reduced granule cell models that they have in their model by the reconstructed models that we constructed based on morphological reconstructions from Norberto's lab. And by doing so, we had to rearrange all the synaptic connections in this structure. We did that, respecting the percentages of connections arriving at each area, at each layer of the model, which as you can see here, proximal dendrites, medial dendrites, distal dendrites, and also not only in reference to the cell itself, but also in reference to the network model. So inner molecular layer, medium molecular layer, outer molecular layer. So we had to be careful to construct the model respecting all the percentages of the connections, of all the synaptic connections existing, and also the performed path input that arrives in the outer molecular layer. So, I mean, the basic idea is that we reconstructed, we used the same model by replacing the granule cells by more complex ones, more realistic ones. And to do so, we used the database of model that was provided by Norberto. So we had a database of 20 control cells and 20 pillow cells. So we had our default model is this one in blue to the left, at least it's my left here, which 100 newborn cells of non-pillow-treated animals. And then we did several simulations introducing different fractions of pillow-treated granule cells in the network. So we had a model with 10% of the granule cells replaced, say the control granule cells replaced by pillow cells, 20% of those cells replaced by pillow cells, and so on and so forth. Since we have different cells in our database, we kind of randomized the construction of those models so that the granule cells were not the same. Each one of them was different from the other ones. So we had lots of different models with different fractions of granule cells, of pillow-treated granule cells, in comparison to the default model, which has newborn control cells. And what we found when we submitted the network to focal stimulation, reproducing the stimulations by Soltes, is not what we expected, I must admit. We observed what you can see here, that and the first figure is the control network, the default network. So it's a dentate network with morphologically reconstructed control cells. When it is receives focal stimulation, and all of this is for 10% sprouting, which is a case which already has some sort of sustained activity in the studies conducted by Soltes, and we observed more or less the same result that he observed in his works. So activity propagates throughout the network, and then it dies out. It doesn't exhibit self-sustained activity. When we replaced 10% of those cells by pelo-treated cells, we observed very little difference. Still, the activity propagated is sort of sustained, was kept active for a longer period, but then it decays. So the network doesn't exhibit sort of self or reduced sustained activity that we expected if those morphologically altered granule cells had some protective effect. The network still is exhibiting hyperexcitability, and the effect is even stronger if you increase the percentage of pelo-treated cells, say to 50% in the network. It becomes even more excited. So the morphologically altered newborn cells are they are contributing to the hyperexcitability of the network, rather than reducing the hyperexcitability as we predicted based on the findings of our single cell modeling. So I'm just getting to the end of this talk. So based on the combination of, say, Soltes' investigations and our simulations, so far we're still carrying on. We are starting to use the larger model, the 1 to 20 scale model that Soltes has constructed. But so far what we can say is that topological alterations in the network, in the structure of the network, are much more important, are much more critical for hyperexcitability, i.e. epilepsy, than alterations in single cell morphologies. Although single cell morphologies although alterations in isolated cell morphologies are observed, they are, at least based on our findings, not as important to epilepsy as the alterations in the structure of the network, which makes sense, in a sense, based on what has been said in those two previous talks. Everyone is stressing the role of the topology, of the network structure of the system. So our findings go in the same direction, saying that for epilepsy, at least, the structure of the network, the structural features of the network, whether they are small word or regular or random, this is critical, at least in the case of the dentate gyrus, for epilepsy, for hyperexcitability. Also, we can say that for single cells, for single granule cells, those morphological alterations observed in newborn cells, they reduce excitability. We don't know whether this is a real effect or could be an artifact of our models, which, as I told you before, they are based on the maximal conductance densities observed in control cells. We're using the same conductances in our morphologically altered models. It's plausible that those conductances change, the maximal conductances change when the animal undergoes the status epilepticus. So we really need to know how they change, what conductances change, what ionic channels change from the control animal to the pelo-treated animal, but we don't know this data, and this may be causing this reduction of excitability of those single cells. But in the network context, the presence of morphologically altered granule cells have little effect, as we have observed in our model. at least in terms of hyperexcitability. And rather the opposite. It seems that as you increase the proportion of morphologically altered granule cells in the network, they contribute, which means they are not critical for hyperexcitability. It seems that the mossy fibers probably is the critical factor. But the alterations in the morphology of the granule cells, which come on top of that, they contribute to the to further increase of the hyperexcitability of the network. But so the kind of major conclusion that I can say out of this is that topological structure, the topological structure of the network is the critical factor, but the morphological alterations at a single cell level, they may have a concurrent effect, which, based on our findings, contribute to the increase of hyperexcitability of the network. So that's basically it. This is the team, people who did this work with me. Julian Tejada, who is not here, from my lab. Now he's a postdoc student. And Norberto and Gabriel, who is here as well. They are the guys who gave us the data, the morphological data, which you used. And the final slide is also very important, is our sponsors. Thank you.